TF network map indicates the direct targets of a DNA binding transcription factor. In traditional computer science, we're taught to think of data as being free and to design analysis algorithms that run as quickly as possible as a function of the size of the data set. In systems biology, on the other hand, we must think of data generation as being part of the algorithm and develop algorithms whose total time, including the time required for data generation, grows both slowly and predictably as a function of the size of the required data set. So it's not sufficient to download a few large data sets, show that an analysis algorithm works well on them, without considering the cost of generating new problem instances that the analysis algorithm applies to. From that perspective, there is good data and bad data. I group among the good data gene expression profiles, including those that are derived from cells in which a single transcription factor has been knocked down or knocked out, and genome sequences, of course. And among the bad, I group transcription factor binding locations as determined by chip experiments. Classic TF network mapping algorithms focus on gene expression data, and more recently there have been published algorithms that are described as being integrative, that make use of all the available data, including the finicky and hard-to-get data. That's not what we want to do. So a few years ago, we developed an algorithm called NetProphet that takes advantage of expression profiles from cells in which a TF has been perturbed. And actually, Michael Kramer, who spoke a few minutes ago, was one of our co-authors on that original paper. And today I want to talk about NetProphet 2, which improves accuracy further by incorporating information that can be extracted from genome sequences. So it's based on three ideas. The first is that combining approaches to analyzing gene expression data improves accuracy. The first NetProphet version used lasso regression of target gene RNA levels on transcription factor RNA levels and combined that with the probability that the target is differentially expressed when the transcription factor is knocked down or knocked out. NetProphet 2 adds another method of predicting target gene expression as a function of transcription factor expression, known as Bayesian additive regression trees, or BART. And while BART alone performs worse than NetProphet 1, the combination of the two performs better. The second idea is that TFs with similar DNA binding domains, meaning the amino acid sequences of the DNA binding domains are similar, tend to bind similar DNA sequences. And this chart simply shows that that's true in yeast. The way we exploit this idea is we start with a score matrix whose rows are transcription factors, columns correspond to target genes, and entries reflect the likelihood that the transcription factor directly regulates the target gene. We then replace each row for a transcription factor by a weighted sum of other rows. And the weights are essentially the normalized percent identity between the DNA binding domains of the two rows. So here I've illustrated how that works for row 1 in this matrix. The last idea is that transcription factor binding specificities can be inferred from score matrices and genes promoter sequences. So for each TF, we identify motifs in promoters that distinguish high-scoring from low-scoring targets using an existing algorithm called FIRE. And then we score all the promoters in the genome for the presence of the motif, producing another score matrix, which we then combine with our other score matrices. And this slide shows a comparison of NetProphet 2 to a number of other algorithms using both chip data as a gold standard and using binding potential determined from in vitro experiments as a gold standard. And we tested it on both yeast and fly. And a few things to notice here are that Genie 3 slightly outperforms NetProphet 2 on the PWM standard in yeast. And CLR performs relatively well on the chip standard in fruit fly. But overall, NetProphet outperforms, and you see that particularly when you look at edges supported by both PWM data and chip data, where NetProphet 2 seems to outperform well. So I'd like to finish by thanking my co-authors Yiming Kang and Hyunha Liao. One question? Yes.
Just with your perspective about the cost of data.
Yes.
Some data also, some data like the PWM is more generalizable than others. Some context specific. So sometimes it might take a long time to generate data, but then it's more valuable because it is applicable more widely. Is that incorporated into your?
Yeah, that's an excellent point. I think that's very important. It's not the binding potential measures are not context or cell type or growth condition dependent, whereas things like chip experiments are. Good point.